Bismillahir Rahmanir Rahim. Welcome again to Derma Immunology. I am Naglaa Elmongy, Professor of Dermatology, Cairo University. Our video today will be about the role of macrophages in wound healing. My reference today will be this review that was published in Cells in 2022 October, and its title is Macrophage Mediated Inflammation in Skin Wound Healing. Wound healing is a complex process but finely tuned. Wound healing starts immediately after injury and may persist for months or even years after the wound is sealed. Wound healing involves a series of interconnected and overlapping physiological stages that include hemostasis, inflammation, proliferation, and remodeling. Wounds are categorized as either acute or chronic. Acute wounds follow a predictable healing rate, while chronic wounds failing to heal within 6 weeks due to inefficient cellular and molecular functions may necessitate limb amputation if untreated in some cases. Approximately 188 proteins have been identified to be overexpressed by more than twofold in chronic wounds, potentially contributing to chronic inflammation, compromised angiogenesis, and decreased cell viability. The initial phase of wound healing is referred to as the inflammatory response, which is the first among several overlapping stages. Inflammation has been noted to delay the healing process and promote heightened scar formation. Macrophages play a key role in regulating inflammation and wound healing. They protect the host through actions like phagocytosis, initiation and resolution of inflammation, as well as releasing growth factors for cell proliferation and tissue recovery in wounds. Macrophages release growth factors like epidermal growth factor, keratinocyte growth factor, and tumor growth factor alpha. These factors promote fibroblasts and keratinocyte proliferation, collagen, and extracellular matrix production, facilitating wound granulation and reepithelialization. Also, macrophages maintain equilibrium between pro-angiogenic and anti-angiogenic signals within wounds, effectively regulating the angiogenic response during tissue granulation and scar resolution. Typically, circulating monocytes move towards the wound site and transform into macrophages. These macrophages remove dead neutrophils through a process known as erythrocytosis. In parallel with monocyte-derived macrophages, resident macrophages also induce inflammatory responses by producing hydrogen peroxide, which attracts blood neutrophils and monocytes. Both migrating monocytes, macrophages, and tissue-resident macrophages are recognized as primary regulatory cells with critical responsibilities in inflammation management. Macrophages have specific functional phenotypes in response to microenvironmental signals, a phenomenon termed macrophage polarization. This polarization either encourages or inhibits the inflammatory phase of wound healing. Macrophages are categorized classically into two groups based on phenotype and function. Classically activated or pro-inflammatory or M1 CD86 positive macrophages, which release cytokines like interleukin 12, interleukin 1 beta, interleukin 6, and tumor necrosis factor alpha, and induce nitric oxide synthase. They play roles in pathogen removal, cytokine release, and promoting T helper 1 type responses. The second group, or the alternatively activated or anti-inflammatory M2 CD206 positive macrophages, foster angiogenesis, repair the extracellular matrix, release anti-inflammatory cytokines, and facilitate inflammation resolution. In this diagram, we can see the main or classical two types of macrophages, which are M1 macrophages and M2 macrophages. M1 functions is a pro-inflammatory and bacterial phagocytic, and they secrete pro-inflammatory cytokines like interleukin 1 beta, tumor necrosis factor, and interleukin 6, also chemokines, and free radicals. The other subtype, which is M2 macrophages, and their functions are anti-inflammatory, pro-angiogenesis, and pro-wound healing. And they secrete anti-inflammatory cytokines like interleukin 10, interleukin 1 receptor A, chemokines, and growth factor like transforming growth factor beta 1, and vascular endothelial growth factors, and others. Macrophage phenotypes shift from M1 to M2 upon engulfing neutrophils, guided by mediators from neutrophils. During the once inflammatory phase, macrophages present polarity between M1 and M2 phenotypes, regulated through cytokines, oxidants, lipids, and growth factors produced by the macrophages themselves. Macrophage plasticity is epigenetically controlled by histone and DNA modifications, as well as microRNAs. Also, interactions with various cells like adipocytes, infiltrating immune cells, neutrophils, and T cells, and keratinocytes affect macrophage polarization. Previous research has demonstrated that macrophages produce mediators with autocrine effects that affect the inflammatory response of M1 phenotype macrophages. The production of interleukin 1 beta and node-like receptor family pyrene domain containing 3 by macrophages are key drivers of inflammation and the M1 phenotype, contributing to the disturbed wound healing regulation. Inflammation caused by macrophages requires two signals: an initial signal for immature interleukin 1 beta transcription and a subsequent danger signal for mature interleukin 1 beta release. Macrophage expressed glycoprotein NMB plays a role in shifting macrophage polarity from the M1 phenotype to the M2 phenotype. Studies have indicated that the absence of Notch signaling leads to elevated levels of tumor necrosis factor alpha, interleukin 6, interleukin 12, and nitric oxide synthase, intensifying inflammation via its impact on the toll-like receptor and nuclear factor kappa B pathways, a phenomenon observed in diabetic situations. Macrophages also generate matrix metalloproteinases, enzymes that break down both matrix and non-matrix proteins. Matrix metalloproteinases play a significant role in altering the phenotype and function of macrophages. For example, macrophages secrete high concentration of matrix metalloproteinase 9 or gelatinase B after invading the wound site. This matrix metalloproteinase is capable of cleaving macrophage integrin beta 2, leading to a shift in macrophage phenotype. However, in vivo research reveals that macrophages present broader characteristics than the simplified M1 M2 phenotypes observed in laboratory settings. So there are diverse macrophage subtypes that express a mix of macrophage-specific markers with distinct origins. Transcriptomic and functional assessments highlighted different rules for dermal macrophages. Specifically, those with elevated phagocytic activity express specific genes such as C4b and others, along with scavenger receptors stabilin 1 and CD36. Dermal wound macrophages consistently change their phenotype from pro-inflammatory to reconstructive. Numerous studies categorize M2 macrophages by their roles in wound healing. These are further divided into three subtypes: M2A, M2B, and M2C. M2A is triggered by interleukin 4 or interleukin 13, leading to elevated secretion of arginase 1, insulin, growth factor 1, and other cytokines by macrophages. M2A macrophages additionally support angiogenesis, fibroblast activities, migration, and differentiation. M2B macrophages balance anti-inflammatory and pro-inflammatory rules by releasing cytokines like tumor necrosis factor alpha, interleukin 6, interleukin 1, as well as the anti-inflammatory ones like interleukin 10 and interleukin 12. As such, this macrophage subtype can be a status between M1 and M2A polarization states. Upon exposure to interleukin-10 transforming growth factor beta or glucocorticoids, M2C macrophages show potent anti-inflammatory effects. Also, they promote angiogenesis by inducing migration and tube formation in high endothelial cells. These macrophages generate matrix metalloproteinases 9 to clear vessels and absorb blood-derived stem cells at injury sites. They also engage in phagocytosis of wound debris and contribute to the deposition of extracellular matrix components. An important point regarding the role of macrophages in wound healing is to know the contribution of tissue-resident macrophages and non-bone marrow-derived macrophages in modulating inflammation and wound healing. However, there is a lack of adequate research investigating the precise contributions of both bone marrow and non-bone marrow-derived macrophages in wound healing process. Prior research has indicated that chemotherapy and radiation can result in bone marrow impairment, causing a delay in hematopoiesis recovery and subsequent monocyte migration into the circulation. So it's very important to investigate whether non-bone marrow derived macrophages can potentially compensate for the delayed migration of circulating monocytes to effectively regulate the wound healing process. In this diagram, we can see the different factors that affect the function of macrophages in wound healing. So we have pathological conditions that can affect macrophages like aging, where there is an increase in the pro-inflammatory M1 phenotype, obesity, where there is a diminished scavenger receptor and erythrocytosis activity in macrophages, and enhanced fibrosis during wound healing. And also we have the problem of diabetes, where we have elevated type 1 macrophages and increased M2 macrophage apoptosis, and also with bacterial infection where we have elevated type 1 macrophages also. The other important factors that affect macrophage function is epigenetics, which include histone methyl transferases, DNA methyl transferases, and histone demethylase. Also, we have cellular interactions which affect macrophage functions, like keratinocytes that produce exosomes, adipocytes that produce adipocytes, lipids like palmitic acid, oleic acid, and others. And we have T cells which can secrete interleukin 17 that push to the inflammatory side, and neutrophils where we have NETs and matrix metalloproteinases 2, 8, and 9. Now I'm going to talk about macrophage metabolism and plasticity in diabetic wounds. Diabetic wounds bring about changes in macrophage metabolism. Macrophages shift their energy source for generating ATP because glucose plays an important role in regulating this ATP production. Actually, glucose provides precursors for histone acetylation and methylation, which are known to be two major epigenetic processes affecting macrophage plasticity and function. Nicotinamide adenine dinucleotide phosphate, or NADPH, is vital for reactive oxygen species production and is produced through glycolysis. Diabetes often disturbs glucose metabolism, causing alterations in macrophages driving cytokines like interleukin 1 beta. This disruption subsequently diminishes glycolysis in macrophages present in diabetic wounds. While the precise cause of reduced glycolytic capacity in macrophages remains unclear, it appears that monocyte-derived macrophages from bone marrow are more susceptible to changes in glucose metabolism than tissue-resident macrophages. Moreover, macrophage alternative activation relies on alternative energy sources like lipid synthesis and the conversion of arginine to proline during the wound healing phases of proliferation and remodeling. Diabetes leads to pro-inflammatory macrophages implicated in various diabetic complications. This shift could impact macrophage metabolism since M1 and M2 phenotypes depend on glycolysis, oxidative phosphorylation, and tricarboxylic acid dependent mitochondria for ATP production. Also, there is a hypothesis that reactive oxygen species induced mitochondrial damage could prevent the transition from M1 to M2 phenotype in macrophages. Also, other researchers showed that improper macrophage functions in diabetes are tied to glucose metabolism. Excess glucose triggers NLRP3 inflammasome overactivation, increasing interleukin 1 beta expression. This results in heightened M1 macrophage induction and elevated production of harmful pro-inflammatory cytokines that delay wound healing in diabetes. Other researchers found that membrane type 1 matrix metalloproteinase promotes macrophage glycolysis through hypoxia-inducible factor activation. This suggests that hypoxia-inducible factor 1 regulates oxygen levels in diabetic ones, affecting macrophage metabolism. Also, in diabetic ones, elevated interleukin 1 beta due to toll-like receptor for overexpression might alter macrophage metabolism. Earlier research indicated that glycolysis primarily powered ATP production in M1 macrophages. However, glycolysis is a fundamental process that supplies ATP for both M1 and M2 macrophages. Therefore, alterations in glycolysis and related factors are likely to affect macrophage functionality and consequently impact the healing of diabetic wounds. And unlike regular wounds that undergo a controlled shift from M1 pro-inflammatory to M2 pro-healing macrophages, diabetic wounds show prolonged M1 polarization. This leads to prolonged inflammation and delayed healing, while the exact changes in macrophage function in diabetes are not fully understood. Connecting disturbed macrophages plasticity only to hyperglycemia is an oversimplification. Some researchers proposed that the cyclooxygenase 2 prostaglandin E2 pathway, a regulator of macrophage driven inflammation, is prominently activated in human and murine wound macrophages. This pathway triggers NF-kappa B activated inflammation in M1 macrophages. A different study highlighted that monocytes experience oxidative stress leading to toll-like receptor 2 induced NF-kappa B activation in diabetic wounds. Further research indicated the significance of vitamin D in suppressing NF-kappa B downstream pathways. This resulted in reduced expression of NF-kappa B driven pro-inflammatory cytokines interleukin 1 beta, interleukin 6, and tumor necrosis factor alpha in diabetic mice. Beyond the NF-kappa B, the pro-inflammatory chemokine ligand 2 has a key role in modulating macrophage function in diabetic wounds. Research has revealed that an inverse relationship between chemokine ligand 2 levels in diabetic wounds and the occurrence of M2-like macrophages. Also, studies have indicated that this chemokine plays a key role in sustaining the presence of M1-like macrophages in wound sites. Now I'm going to talk about factors affecting macrophage activity. Throughout the wound healing process, Macrophage phenotype is controlled by epigenetic alterations such as histone modification and DNA modifications, microRNA functions, ATP-dependent remodeling, and interactions with other cells. Epigenetic controllers play a key role in skin wound healing by dynamically regulating cell proliferation and migration of keratinocytes and endothelial cells. Additionally, research has demonstrated that epigenetic elements regulate macrophage biology through complex mechanisms to modulate gene activation, transiently reshaping cellular phenotype and function. Histone modifications are significant contributors to altering macrophage phenotypes throughout wound healing. DNA methylation is linked to macrophage plasticity, and microRNA have the capacity to affect macrophage behavior in diabetic wound situations. I will not go deeply in this video in details about epigenetic regulation of macrophage. Uh, However, if you want to get more information, you can read the reference that I have just mentioned in the beginning of this video. And here is also another recent reference which is published in Burns Trauma 2023 January, and its title is Epigenetic Regulation of Macrophage Polarization in Wound Healing. Macrophage function is closely tied to epigenomic organization. Transcription factors are vital for macrophage polarization, while epigenetic changes to these factors shape macrophage heterogeneity. On the contrary, transcription factors itself regulate epigenetic enzyme expression, forming an interdependent network that controls macrophage plasticity. Macrophage phenotype can also be affected by the wound microenvironment. Changes in the phenotypes of keratinocytes, adipocytes, T cells, and neutrophils have been observed in diabetic wounds, potentially impacting their interactions with macrophages in the wound site. Dermal adipocytes have the ability to generate palmitic acid, oleic acid, monocyte, chemoattractant protein 1, and tumor necrosis factor. These biomolecules produced by adipocytes can affect the inflammatory phenotype of macrophages. The changes caused by obesity in macrophages and adipocytes result in persistent inflammation and insulin resistance. Insulin resistance associated with obesity has been linked to increased levels of pro-inflammatory cytokines like tumor necrosis factor alpha, interleukin 1 beta, and interleukin 6. Adipocytes release these cytokines as a result of heightened pro-inflammatory factor release during obesity progression. Factors like free fatty acids, triglycerides, resistin, leptin, retinol binding protein 4, interleukin 6, tumor necrosis factor alpha, and interleukin 1 beta contribute to this. Consequently, adipocytes can exert a significant role on macrophage-mediated skin wound healing. Keratinocytes also produce various cytokines and chemokines and have a significant role in cutaneous immunity. In cases of chronic wound inflammation, keratinocytes modulate NF-kappa B to release cytokines and interferons, thereby affecting the inflammatory profiles of immune cells. In addition, chronic wound edges show heightened keratinocyte proliferation compared to regular ones. And in one study, they found that chemokine ligand 2 released by keratinocytes leads to macrophage trafficking and the production of epidermal growth factor by macrophages in the wound. Macrophage released epidermal growth factor will in turn stimulates keratinocyte proliferation. In another study, they found exosome-mediated communication between keratinocytes and macrophages during skin wound healing. The study established that keratinocytes released exosomes affect macrophage plasticity. Blocking keratinocyte-derived exosomes in vivo led to a significant rise in the pro-inflammatory macrophages within skin wounds. This highlights that the behavior and role of wound bed macrophages can be affected by various cell types, particularly keratinocytes. In the wound healing process, immune cells that enter the wound site have been demonstrated to affect macrophage phenotype. Neutrophil, the initial cells to arrive at the wound, release neutrophil extracellular traps or nets. Diabetic wounds show significantly elevated levels of nets compared to healthy wounds. Neutrophil extracellular traps or nets trigger inflammation and induce macrophages to secrete interleukin 1 beta. Also, heightened neutrophil levels, serine elastase, and inflammatory macrophages contribute to increased growth factors and protease activity, example matrix metalloproteinases 2, 8, and 9, resulting in prolonged inflammation. Lymphocytes play an important role in macrophage polarization. Increased number of T cells, including gamma delta and T helper 17 cells, have been observed in diabetic wounds. T helper 17 cells release interleukin 17, which regulates macrophage polarization. Eliminating interleukin 17 improves wound healing in diabetic mice by reducing M1 macrophages and enhancing M2 macrophages. In summary, macrophages are vital immune cells that react to infections and modulate pathophysiological processes like wound healing. Through their phagocytic activities and the release of cytokines and growth factors, macrophages play a key role in coordinating inflammation, fibrosis, and wound recovery. Macrophages orchestrate wound healing by shifting from primarily pro-inflammatory M1-like phenotypes present in the early post-injury phase to anti-inflammatory M2-like phenotypes that appear later to regulate skin repair and wound closure. Macrophages are key players in skin wound healing, and a better understanding of how macrophages function in wounds can lead to improved therapeutic strategies for promoting skin wound healing. I hope that you have enjoyed this video, and I'm reminding you again to subscribe to the channel and activating the alarming bell, and thank you.